Hello everyone. So welcome to this recorded video presentation on DNA replication. So as you watch, I encourage you to listen carefully and take down notes to help you reinforce your understanding of this important topic. So with this topic, DNA replication, this is an essential biological process that ensures the accurate duplication of genetic material before the cell divides. So this process is vital for growth, development, and repair in all organisms. So let's dive into step-by-step mechanisms of what is DNA replication is all about. Okay, so let's start. So what is DNA replication? So DNA replication class, it is the process of creating two identical copies of DNA from a single original DNA molecule. So it occurs in all living organisms and is a vital part of biological inheritance. Meaning class, DNA replication, it is a process of forming new synthesized DNA molecule from the parent strand. So as you can see on the picture, so the babaw nga part, that is the parent strand. So at the end of DNA replication, along its steps, we can produce another duplicated strand of DNA. Before we dive into the step-by-step mechanisms of DNA replication, let's first understand what are enzymes and why they play a crucial role in this process. Enzymes class are basically the biological catalyst, meaning these are proteins that speed up the rate of chemical reaction in the body without being consumed in the process. So in DNA replication, enzymes play a vital role in unwinding, copying, and sealing the DNA strands to ensure accurate duplication. So you will learn later what are the different enzymes involved in DNA replication. Without these enzymes class, the DNA replication would be too slow and also prone to errors, which could lead to genetic mutations and cell malfunction. Here are the several key enzymes involved and work together in a coordinated manner to ensure the smooth progression of DNA replication. So along the steps of DNA replication, you will learn each what are the functions of these enzymes. Here's the steps in DNA replication. It started with replication fork formation, then primer binding, next elongation process, and lastly, the termination process. So let's explore the processes in detail. To begin with, we have the replication fork formation. So in this process, this is the start of the replication wherein an enzyme called helicase will unwind and separates the double-stranded DNA by breaking the hydrogen bonds between the nitrogenous bases. So this creates a structure called the replication fork where replication will actively occur. For a better understanding of this process, class imagine a zipper being unzipped. So just like a zipper pulls apart towards sides of fabric, helicase also class separates the two strands of DNA to allow the replication machinery to access the template strands. That is for replication fork formation. Next is primer binding. Since na-unwind na ni helicase ang two strands, now primer binding will follow. So the enzyme primase class will lay down short RNA primers which will serve as the starting points for DNA synthesis. So kung lantawon nyo sa strands class, once mag-bind na ang DNA primase, the DNA primase will generate an RNA primer. Once may ara na RNA primer, then pwede na ta ka-start sa elongation process which is the third step. In elongation process class, this is the step in DNA replication wherein there is an addition of nucleotides to the DNA strand. Because of this, we need an enzyme nga mag-add sang nucleotides, and these enzymes class are called DNA polymerase. As soon as the DNA polymerase will bind with the RNA primers, the DNA polymerase will extend the new strand by adding complementary nucleotides in the 5' to 3' direction. So going back to the complementary base pairing, adenine will be paired to T or thymine, then thymine vice versa will be paired with adenine. Same as to guanine to cytosine and cytosine to guanine. Since DNA polymerase can only synthesize DNA in the 5' to 3' direction, replication will occur differently on the two strands. So there are two strands that will be synthesized, the leading strand and the lagging strand. For the leading strand synthesis, the leading strand is synthesized continuously in the direction of the replication fork. So the DNA polymerase will move smoothly along the template strand, adding nucleotides without interruption. Thus, the leading strand acts continuously, unlike The lagging strand class is synthesized discontinuously in the opposite direction of the replication fork movement. So the DNA polymerase creates short fragments of DNA which is known as Okazaki fragments. And class, to help you determine the lagging strand, mabalaan nyo na siya if lagging strand siya, if it moves from 5' or 3' to 5' direction. Now, the lagging strand also, class, ga-exist na siya if the parent strand has multiple RNA primers. And that's it for the elongation process. So once the DNA polymerase naka-add na nucleotides, then we will proceed to the termination process. Once the replication is complete, another enzymes will follow. So it's the exonuclease. The enzyme exonuclease will remove the RNA primers and replaces them with the DNA molecules or nucleotides. And also, the exonuclease class will play a crucial role in proofreading and removing any incorrectly paired nucleotides, ensuring the accuracy of DNA replication. So, for example, kung sa DNA strand, ang parent DNA may adenine, tapos si DNA polymerase, gimbutangan niya nga C, then si exonuclease, i-correct niya na that instead of cytosine, ang hihatag niya gid is si thymine. On the other hand, the DNA ligase class will seal the gaps between the Okazaki fragments, forming a continuous strands. So this process ensures that both strands are accurately replicated and that the DNA molecules is complete. However, at the ends of linear chromosomes, a unique problem will arise. So the DNA polymerase is unable to fully replicate the extreme ends of the lagging strand, leading to gradual shortening of chromosomes over successive replications. So to prevent the loss of essential genetic information, the enzyme telomerase will extend the ends of chromosomes by adding repetitive DNA sequence to the telomeres, which act as protective caps. The telomerase is highly active in germ cells and stem cells but has limited activity in most somatic cells, which is why telomere shortening is associated with aging and cellular senescence. The final result of termination is two identical DNA molecules, each containing one original and only newly synthesized strand, wherein exonuclease will ensure the accuracy, the DNA ligase will seal any remaining gaps from the lagging strands, and also the telomerase will maintain the chromosome stability in certain all types. The importance of DNA replication, it ensures that each new cell receives an exact copy of the genetic information. Also, it allows the proper growth, repair, and reproduction in living organisms. It also maintains genetic stability from one generation to the next. The exonuclease ensures high fidelity in DNA replication by removing errors. The DNA ligase will maintain structural integrity by sealing the gaps in the DNA. And lastly, the telomerase activity will help preserve chromosomes integrity in rapidly dividing cells. In summary, this DNA replication process is a highly precise and efficient process that ensures genetic continuity. So from the replication fork formation to the termination process, multiple enzymes work together to accurately copy the DNA, preserving life's blueprint across generations, and also understanding DNA replication is fundamental in genetics, medicine, and biotechnology. Before I will end this, so please take time to read and answer the following questions and also write it in a 1/4 sheet of paper. Submit this on our first meeting this week. Thank you for listening, and if you have any questions, just feel free to clarify on our class GC. And also class, for our next topic, we will have protein synthesis.